Why you need this supplement on a high-protein diet. Biotin's game-changing effects explained by a PhD. If you are eating a high-protein diet, you probably are not getting enough biotin, and this is especially true if you are a protein-fueled athlete and any of the following apply. Maybe you find your skin too oily, too itchy, or too red. If your blood sugar is getting out of control, or if you just feel down in the dumps, you're tired, you're lethargic, you feel a little depressed. And this is especially true if there's something more serious, like your hands and feet start tingling, or you get subtle sensations of something crawling on your skin. Alternatively, if you're pregnant or looking to conceive, this is something you really need to understand, almost regardless of how much protein you eat. Nevertheless, most biotin supplements on the market have doses that are actually too high. In fact, I myself gave my own self several weeks of clumsiness, short-term memory loss, and short temper by taking 10 milligrams per day of biotin for weeks. And this is a pretty normal dose out there. So most people who supplement with biotin are actually taking these high-dose supplements and are getting too much. And then almost everyone else is not getting enough. And if you're eating a high-protein diet, all the more with the extra stresses of athletic activity or pregnancy, but really just the high protein diet alone will push you from not getting enough, meaning not getting enough to be optimal, into a totally different territory of dangerously low. And this is not something that you want to put off fixing, because not matching your protein with sufficient biotin actually leads to toxic byproducts of branched chain amino acids that will be causing slow and steady neurological decline over time, even if they're not causing any overt problems acutely. Meaning, if you're worried about your cognitive health over the long haul, you really want to be minimizing any of these toxic byproducts, because you don't necessarily know that you're causing a problem to your long-term brain health just by how you feel at the moment. And then that just brings up, like, what are people thinking about when they detox, right? What's the point of taking all these detox supplements, getting lymph drainage massages and hitting up the sauna to work up a sweat, when the protein to biotin ratio of your diet is literally generating new toxins every single time you eat? So in this episode, you're gonna learn about how much biotin you need for a given protein intake, how much biotin you need for these other stressors, especially pregnancy, and how to get that biotin requirement from food and supplements. This episode is only available for 48 hours for free. After that, it's reserved for MasterPass members. So check out the link in the description for how you can get access to the video, the podcast, and the written and fully referenced article version forever, or for as long as you're a member. So this is what you're gonna learn about in this episode. You're gonna find out what biotin is, why no one seems to care about biotin, yet why everyone should, why pregnant women especially should care. We're gonna learn a bit about the basics of what biotin does, the biotin sparing effect of different proteins leading to the protein paradox. So the protein paradox is the fact that biotin raises your need for protein, and yet there are proteins that spare some of the effects of biotin. And this can be a little bit confusing, but we're gonna dig down into it and explain it all. We're gonna look at the best markers of biotin status, the mysterious repetition of 30 and 300 micrograms in the various alternations of doses that we need claimed by different government agencies. We're gonna look at how much biotin we actually need based on the research and according to these different stressors, especially protein. So we'll express that requirement specifically as a function of protein intake, and then look at how it increases in pregnancy and lactation. And we're gonna ask whether, where do you draw the line between what's truly deficient and what's, biochemically suboptimal based on a marker? And some people would say that if you don't have any symptoms, you're fine. But as I said in the beginning, you can not have any symptoms, but you can have a toxic load in your body that is slowly degrading your brain health over time. So I'm gonna make the case that it doesn't matter whether they were deficient by some kind of symptomatic criteria. If you're not biochemically optimized, you're not biochemically optimized, and that will hurt your longevity. Your microbiome, why it won't save you. We're gonna look at the best biotin-rich foods, the so-called biotin rule of thumb. This is my rule of thumb for just how to really simplify, match this food to that food, in several sets of different types of foods. We're going to talk about egg whites as the enemy of biotin and the relationship to protein supplements. And then we're going to get down to the bottom line. So if this is TLDR for you, or TLDW if you're watching, or TLDL if you're listening, just scroll ahead to the end and you can get to the bottom line. And then as an add-on to this, one thing you'll find in the article that I'm not going to go into the video because it's just too technical, we're going to talk about in the article, so see the link in the description if you want the article, why the whole concept of databases of biotin in foods is a serious methodological crisis and why almost none of the numbers can be trusted. So, nevertheless, let's get on to introducing biotin. Biotin is a water-soluble B vitamin that was once known as vitamin H and is now known as vitamin B7, although almost no one calls it either of those things, they call it biotin. The most well-established abundant sources are liver and egg yolks, as well as nutritional yeast. Newer research suggests that it may be very abundant in natto and other fermented foods, and that pasture raising may be important for its content in animal products. In the world of supplements, it is widely regarded as a boost for the health of the skin, hair, and nails. In medicine, its classical deficiency signs are scaly, red, itchy, usually candida-infected rashes around the nostrils and mouth and between the anus and genitals and in any of the body folds, conjunctivitis, hair loss, depression, lethargy and loss of appetite, and tingling and numbness in the extremities, or a sense of something crawling on the skin, various types of paresthesia. In infants and children, it leads to developmental delay, and in most severe cases, it leads to hallucination and seizures, and in babies, floppy baby syndrome. In those with a genetic disorder in its metabolism, untreated deficiency can lead to death. Research also suggests biotin is necessary to prevent birth defects and may be limiting in a large number of pregnant women, and that it should help stabilize blood sugar. And yet, in the mainstream, no one really cares. Biotin. No one cares. Biotin is so universally regarded as irrelevant to modern nutrition that when I looked in 2019, the USDA had no entry for it in its food database. At some point since then, it added a rather pathetic 3-page list of entries that doesn't even include the main dietary sources. When I was in graduate school, my department head was teaching my macronutrient metabolism class. I looked at the biochemistry of biotin when he was teaching it, and I said, couldn't diabetes just be a biotin deficiency? It looks like that, he replied, but there's no evidence of anyone in modern society having a biotin deficiency. It's made by the intestinal bacteria, so we probably don't even need to eat it. In that vein, a 1968 case report of human biotin deficiency began with the sentence, quote, "Biotin is so widely distributed in foods and so abundantly produced by intestinal bacteria that doubt has been expressed concerning the occurrence of a deficiency state in man on the basis of inadequate dietary intake." End quote. It's generally assumed that in order for your diet to make you deficient, it has to contain large amounts of raw egg white. Egg white contains avidin, a protein that binds to biotin, and typical cooking methods destroy a lot of it, but not all of it. The older biotin literature refers to deficiency as, quote, egg white injury. The Food and Nutrition Board, or FNB, of the Institute of Medicine, the IOM, now called the National Academy of Medicine, had so little interest in whether adults might need more biotin that they defined our adequate intakes based on the average intakes of babies. When the FNB has good evidence of how much of a nutrient we need, they set a recommended dietary allowance, or RDA. This is the intake that should cover the needs of 97.5% of people. When they do not have such evidence, they set an adequate intake, or AI. This is the average intake of people who apparently get enough. Usually when they set an AI, they use the mean or median intake of the population. If they had done that, the AI for biotin would be 40 to 60 micrograms per day. In 1998, however, they took the average intake for infants, adjusted it upwards based on body weight, and set the adult AI at 30 micrograms per day. Part of their justification for doing this was that a paper from 1997 supposedly found this dose to be adequate to normalize the excretion of 3-hydroxyisovaleric acid, a marker of biotin deficiency. In their words, this value should be adequate for maintaining normal excretion of 3-hydroxyisovaleric acid in adults. This argument is both circular and wrong. It's circular because that paper defined the normal range as whatever was excreted by the people who had not been made experimentally deficient. All they showed was that the baseline values in their study of 11 people were similar to the baseline values in a different study of 10 people. They provided no evidence whatsoever that the marker was adequately suppressed in these people, and they never claimed that baseline intakes were adequate. It's wrong because the researchers who wrote that paper did not control the biotin intake leading to the normal excretion of the marker. They simply found that subjects eating their normal diet and given an approximately biotin-free multivitamin for 10 days had similarly normal excretion of the marker as other people eating what they usually eat. The FMB themselves said that the average biotin intake was between 40 to 60 micrograms per day. The authors of that study cited a range of 30 to 70 at one point, and elsewhere in the same paper stated that the average intake was thought to be 70. If the subjects were eating what they usually eat, then they were eating something closer to the usual intakes of 30 to 70 or 40 to 60 and did not have an average intake of 30. The spirit of dismissal imbuing all of this was so acutely felt in the world of biotin research that a 1999 editorial in the American Journal of Clinical Nutrition that was trying to explain why we should bother getting good data on the biotin requirement was titled "Biotin Bioavailability and Estimated Average Requirement. Why bother?" Let's move beyond this systematic spirit of dismissal and open ourselves up to the possibility that many people could use more biotin than they get. Now, why pregnant women, as well as others, should care. At minimum, pregnant women should care about biotin. 50% of them spontaneously develop markers of biotin deficiency during pregnancy. Animal experiments suggest that the level of biotin deficiency seen in many pregnant women contributes to birth defects. Smokers should care because smoking accelerates biotin turnover and raises markers of deficiency. Those using the anticonvulsant valproate should care because it prevents biotin recycling and causes deficiency. Other people that definitely need to care about biotin are those with biotin-responsive genetic defects in each of the 5 biotin-dependent enzymes, or in the enzymes that transport and recycle biotin, or the enzyme that incorporates it into enzymes as a cofactor. And my estimation is that approximately 1 in 30 are at least carriers for one of these and at least have some major increase in their biotin requirement into the milligram range as a result. People who eat egg whites should care because egg whites contain avidin, a protein that binds to biotin, and typical cooking methods destroy a lot of it, but not all of it. I will make the case below that the rest of us should also care. But first, some biochemistry. This is what biotin does. If you need more biochemistry background to understand this section, I highly recommend my energy metabolism course. So click on the link in the description. You can go to the written version of this article and you'll see courses in the menu and you can just click on energy metabolism. Biotin is one of 2 vitamins used to carboxylate molecules. The other is vitamin K. Carboxyl groups are molecules of carbon dioxide that have been attached to other molecules. So to carboxylate a molecule is to add carbon dioxide to it. Vitamin K uses carbon dioxide directly, while biotin uses carbon dioxide that has been dissolved in cellular water to form bicarbonate, essentially the dissolved form of baking soda. The enzymes that do this are called carboxylases. There are 5 carboxylases that are dependent on biotin. The first one is propionyl-CoA carboxylase. This enzyme forms methylmalonyl-CoA from fatty acids with odd numbers of carbons and from the amino acids isoleucine, methionine, and valine. Methylmalonyl-CoA is subsequently converted to succinyl-CoA using a vitamin B12-dependent enzyme, which can then enter the citric acid cycle to contribute to energy metabolism. Methylmalonic acid, which is what joins coenzyme A to become methylmalonyl-CoA, is used as an inverse marker of vitamin B12 status. Since biotin is needed to make it, a biotin deficiency could mask a B12 deficiency. Acetyl-CoA carboxylases 1 and 2. This enzyme has 2 forms that are used together to coordinate the shift away from fatty acid oxidation in the fasting state toward fatty acid synthesis in the fed state. One of them plays a regulatory role to shut off the carnitine shuttle in response to insulin and high energy status, which stops fatty acids from being imported into the mitochondria to be burned for energy. The other plays a direct role in fatty acid synthesis. This is not just used to make new fatty acids, but it is also used to make existing fatty acids longer, such as in the conversion of the essential fatty acids in plant oils to the forms found in animal products like fish, liver, and egg yolks. And it's these longer forms that are needed by our bodies to prevent essential fatty acid deficiency. Beta-methylcrotoneal-CoA carboxylase. This enzyme is needed to burn the amino acid leucine for energy. Leucine is broken down in several steps to form beta-methylcrotoneal-CoA. Biotin is used to add carbon dioxide to it, thereby forming beta-methylgluconyl-CoA, which can be further broken down into the ketone body acetoacetate or into acetyl-CoA, both of which can then be burned for energy using the citric acid cycle. And fifth and finally, pyruvate carboxylase. This enzyme plays two roles in glucose metabolism. First, when you burn glucose for energy, you break it into two molecules of pyruvate. Pyruvate can then be decarboxylated to form acetate, which is attached to CoA to form acetyl-CoA, or it can be carboxylated to form oxaloacetate. Oxaloacetate is kindling for the citric acid cycle, which is the metabolic flame in which all food energy is burned. The second role is in gluconeogenesis. Lactate and certain amino acids generate pyruvate, and several rewirings allow glycolysis to run in reverse. The first rewiring is the conversion of pyruvate to oxaloacetate, which is then sent off into reverse glycolysis rather than used for the citric acid cycle. In addition to these biotin-dependent enzymes, there are several other enzymes important to biotin metabolism. Holocarboxylase synthetase adds biotin to the amino acid lysine within the biotin-dependent enzymes above. This is an ATP-dependent step. It also adds, this as well as everything I described above is ATP-dependent steps. It also adds biotin to proteins called histones that coordinate DNA inside the nucleus. This plays a role in regulating gene expression. When biotin-dependent enzymes are digested, both within our cells and within our gastrointestinal tract, the biotin is released bound to the lysine. This complex is known as biositin. The enzyme biotinidase is needed to free the biotin from the lysine. Thus, biotinidase is important both to the extraction of biotin from foods and to the recycling of biotin within the body. Foods have some free biotin as well as some biositin or biotin-bound protein that releases biositin during digestion. Once all of the biotin is freed, it has to be absorbed by the sodium-dependent multivitamin transporter, which also helps transport it inside the body in the kidney, preventing it from being lost in the urine. In brief, without biotin, you cannot break leucine down for energy. You cannot synthesize fatty acids. You cannot convert plant oil fatty acids into the forms of essential fatty acids you need. You cannot make new glucose when you need it. You cannot store glucose as fat, and you cannot properly burn it for energy. If you are B12 deficient, you may not know it if using methylmalonic acid as a marker. That you can neither store glucose as fat nor burn it for energy is what prompted my grad school era question of whether this could play a role in diabetes. This would presumably cause devastating effects to glucose disposal. The usually candida-infected skin rash is wildly similar to that seen in deficiencies of riboflavin, B6, and essential fatty acids. In rats, linoleic acid protects against it. And in humans with biotin-responsive genetic disorders who are losing their hair, even before biotin is supplied, a mixture of essential fatty acids that includes arachidonic acid can make the hair grow back. So, what would explain this? Well, in my view, it is the fact that riboflavin is needed to convert the plant form of B6 pyridoxine into the animal form, the one we need, pyridoxal, which is needed together with biotin to convert linoleic acid to arachidonic acid. Arachidonic acid in turn is needed to make prostaglandin E2, also known as PGE2, which signals the formation of tight junctions between skin cells. If any links are missing in the chain that converges on PGE2 synthesis, the epithelial barriers open up. The skin becomes permeable to water, which evaporates, leaving the skin dry. Any microbes on the surface of the skin take opportunity to invade, and they infect the lesion. Linoleic acid probably helps by simply stuffing the low-functioning enzymes with enough substrate to push forward some arachidonic acid. And in humans, showing that the cutaneous manifestations can be reversed, I think, is simply a matter of actually supplying arachidonic acid in the mixed oil that they've used. The most effective way to bypass this entire scheme would be to eat liver and egg yolks. They not only are the most well-established sources of biotin apart from nutritional yeast, but they bypass the entire need to convert linoleic acid to arachidonic acid because they are also the best sources of arachidonic acid itself, and that only applies to liver and egg yolks and does not apply to nutritional yeast. Research from genetic defects in pyruvate carboxylase deficiency suggests that defective fat synthesis is compromised that it compromises the synthesis of myelin and can lead to widespread demyelination. I would also point out that glucose metabolism through the citric acid cycle is the main source of glutamate and GABA, and that energy in the form of ATP is needed for the regulation of all the neurotransmitters. So I suspect that the neurological consequences of short-term deficiencies that can be easily recovered from are due to neurotransmitter defects, and that such consequences of long-term deficiencies that are harder to recover from are a result of demyelination. But I would also add on to this that going beyond pyruvate carboxylase deficiency, you're also compromising the breakdown of branched chain amino acids and some of the other amino acids. And when you compromise amino acid breakdown, you wind up with toxic intermediates of the metabolic pathways that are, generally speaking, neurological toxins. And if they're not causing an acute neurological disorder, that does not mean that they're not leading to long-term neurological damage underneath the hood. Presumably, consumption of organ meats, especially disease-free brain, would help compensate for the defects in the synthesis of many different lipids. Looking at glucose alone, biotin deficiency should predispose to hypoglycemic episodes in the fasting state and to glucose intolerance in the fed state. Nevertheless, there is an escape valve: protein. Protein will not help much with the hypoglycemia because gluconeogenesis from protein requires pyruvate carboxylase. It should, however, blunt the blood sugar rush that comes with eating carbs. Normally, glucose provides everything needed for its own metabolism. If there is enough oxaloacetate, you turn pyruvate into acetyl-CoA and burn it for energy. If there is not, you use biotin to make more. In the biotin deficient state, however, protein can keep the citric acid cycle going. The amino acids asparagine and aspartate are alternative sources of oxaloacetate. Arginine, histidine, proline, glutamine, and glutamate can feed in as alpha-ketoglutarate. Tyrosine and phenylalanine can feed in as fumarate. These will all help generate oxaloacetate that can bring acetyl-CoA from glucose into the citric acid cycle to be burned for energy. Thus, protein should prevent glucose intolerance in the biotin deficient state. However, it's also possible that because in the biotin deficient state you are relying more on these amino acids just barely filling up the citric acid cycle, that the overspilling of the citric acid cycle, which is usually playing a role in the amplification of insulin release by protein response to carbohydrate, you may actually require more protein to do that. So protein might be your best protection against glucose intolerance in the biotin deficient state, but you may need more protein than usual in that biotin deficient state because now the role of protein in allowing you to release insulin is basically protein has to work overtime to do the job of carbohydrate and to do the job of protein. And that's despite the fact that one of the main points of this episode is that protein raises your requirement for biotin. So it's not actually a solution, it's just a stopgap. And if you're burning through your protein in order to do this, while you are also raising your biotin requirement, you are likely to be progressing into a more and more biotin deficient state over time. Now, if we circle back to the neurological issues, protein will probably also protect against neurotransmitter imbalances by keeping the citric acid cycle running. Let's go a little bit more in depth into biotin and protein. Talk about the biotin sparing effect of different proteins, meaning the different amino acids that are present in different proteins. So I analyzed the ratio of biotin sparing to biotin requiring amino acids in a handful of different proteins. In the written version, the spreadsheet is included. Selected based on the convenience of the comparative tables provided in the source that I linked to in that spreadsheet. For this analysis, leucine, isoleucine, methionine, and valine were considered biotin-requiring amino acids. Amino acids that generate acetyl-CoA or pyruvate but have no alternative routes into the citric acid cycle were considered neutral. While they do depend on biotin because acetyl-CoA must combine with oxaloacetate, pyruvate must be converted to acetyl-CoA or oxaloacetate, there's nothing you can replace them with to change this. Replace them with fat, you get acetyl-CoA. Replace them with carbs, you get pyruvate and acetyl-CoA. Thus, they do require biotin, but they are substantially equivalent to carbohydrate and roughly equivalent to fat. Amino acids that can enter the citric acid cycle by any means that does not depend on biotin were considered biotin sparing. These included arginine, histidine, glutamine, glutamate, proline, tyrosine, phenylalanine, asparagine, and aspartate. The ratio of biotin sparing to biotin requiring amino acids gives the biotin sparing index, which I created. By far and away, the most biotin sparing protein is gelatin. Its biotin sparing index is 6.5. Most plant proteins were between 2.3 and 2.8. However, potato stood out at 3.9 and wheat flour stood out at 4.3. Notably, these foods are loaded with carbohydrates, so if you're trying to protect against glucose intolerance, you would need to use isolated plant proteins and not the whole foods. Most animal proteins were similar to, but slightly lower than, most plant proteins, ranging from 2.1 to 2.4. Apart from gelatin, pig milk stood out with 3.4, but no one uses that. Whey protein was typical at 2.3. Overall, plant proteins are slightly more sparing than animal proteins, but whole plant proteins, or whole plant foods, have far less protein and more carbohydrate than whole animal foods, so they're impractical to use. Any whole food animal protein or any protein isolate from animal or plants is likely to work, and gelatin would appear to be the best. Of course, I'm not suggesting anyone avoid fixing a biotin deficiency for any other reason than science. Rather, I am simply making the point that someone in a biotin deficient state who is consuming more protein is less likely to suffer from glucose intolerance and neurological problems than someone in the same state consuming less protein. Further, I believe we should study protein-responsive glucose intolerance as a potential index of biotin deficiency. Biotin and the protein paradox. Based on the biochemistry, protein should be an escape valve that helps prevent glucose intolerance and normalize neurotransmitters, but it should hardly fix the underlying problem. First, biotin deficiency compromises the ability of leucine, isoleucine, methionine, and valine to enter the citric acid cycle by creating deficient activities of propionyl-CoA and beta-methylcrotenyl-CoA carboxylases. It also compromises the ability of amino acids that are metabolized to acetyl-CoA, such as tryptophan and lysine, or to pyruvate, such as alanine, cysteine, serine, and glycine, and threonine, to enter the citric acid cycle by preventing pyruvate carboxylase from making oxaloacetate. Second, when beta-methylcrotenyl-CoA carboxylase activity is missing, beta-methylcrotenyl-CoA borrows a different enzyme from fatty acid oxidation to assist in its metabolism. This enzyme is enoyl-CoA hydratase, which converts it to 3-hydroxyisobutyryl-CoA. Most likely, this inhibits fatty acid oxidation to some degree, which might predispose to weight gain, fatigue, fatty liver, or hypoglycemia. The reason that it would contribute to hypoglycemia is that while protein provides the building blocks for gluconeogenesis, fatty acid oxidation provides about half the energy in the form of ATP. Third, all of these incompletely broken down molecules are toxic. And this is what I was saying at the outset. You can do your lymph drainage, your sauna, your detox supplements. If you're generating toxic molecules from the food that you eat, what's the point, right? So first, focus on optimizing your biochemistry so that you're not spilling out metabolic toxins from the nutrients that you're trying to metabolize. Whenever an energy metabolism pathway cannot be completed, the intermediates remain bound to CoA. This starves cellular energy metabolism, which depends on a robust supply of free CoA. Essentially, this is like passengers who refuse to get off of a bus. If enough passengers refuse to get off enough buses, they can shut the whole city's public transportation down. Not only that, but many of the intermediates inhibit important enzymes. The most well-characterized example of this is the urea cycle. The urea cycle is how you get rid of ammonia from protein metabolism. Acetyl-CoA is needed to activate this cycle by forming N-acetylglutamate, which acts as the cycle's on switch. If a blocked metabolic pathway is starving the free CoA pool, you cannot make acetyl-CoA for this process. Worse, many of the small intermediates will resemble acetyl-CoA sufficiently to act as the wrong key that gets stuck in the keyhole, and through a process known as competitive inhibition, prevent what little acetyl-CoA is available from being used. This double whammy puts a brake on the urea cycle and causes ammonia levels to rise. This can cause bad breath and fatigue when it's moderate, or coma and death when it's very severe. The cell will do its best to defend the free CoA pool by transferring these intermediates to glycine or carnitine. That's why many metabolic defects are diagnosed with the acylcarnitine profiles in plasma or acylglycine profiles in urine. These are profiles of all the different intermediates that could be detoxified with carnitine or glycine. But this also means that you're losing carnitine and glycine. The loss of carnitine is another whack at fatty acid metabolism, and the loss of glycine can have all sorts of negative effects on bones, blood sugar, sleep, mood, mental health, muscle tension, and can, even if your glycine levels get too low, cause you to get startled all the time. The protein paradox is this. While protein is likely to protect against hyperglycemia and some of the neurological consequences of biotin deficiency, it raises your need for biotin by increasing the biotin-dependent oxidation of branched-chain amino acids, or BCAAs, which are leucine, isoleucine, and valine, and methionine. Now, this is where being an athlete comes in, because when you are engaging in athletic activity, you are increasing the muscle metabolism of the branched-chain amino acids and putting even more stress on this pathway. You may be wondering, how did I just call protein biotin sparing if I'm now saying it raises the need for it? The issue is that the biotin sparing amino acids will help you burn carbohydrate for energy, but they will not help you burn the biotin requiring amino acids for energy at all. That's because there are two separate biochemical problems. The limitation on carbohydrate metabolism is the pyruvate carboxylase reaction, and the biotin sparing amino acids completely get around that reaction by supplying alternative sources of oxaloacetate. By contrast, the limitation with the other two carboxylases is that you cannot clear propionyl-CoA or beta-methylcrotenyl-CoA, and those reactions are outside the citric acid cycle. So firing up that cycle's flame does not help that problem. Since the backup in these pathways is toxic to energy metabolism, pushing protein through them without enough biotin creates its own unique problem. So for most proteins, every 40 grams of help that you give to glucose metabolism in the citric acid cycle gives you 20 grams of unrelated collateral damage, and they don't average or cancel each other out. The exception is gelatin, and by extension, collagen or any collagen-based food protein, such as bone broth, gelatin, etc. For every 40 grams of help, it only causes 6 grams of collateral damage. Now, you may have another question. You might look at the 5 enzymes and ask why carbohydrate through pyruvate carboxylase or acetyl-CoA carboxylase, or odd-chain fatty acids through propionyl-CoA carboxylase wouldn't increase the need for biotin in the same way. The reason is as follows. 1, the pyruvate carboxylase reaction is driven by demand, not supply. A low protein diet is likely to increase demand for pyruvate carboxylase, but all things being equal, a high carbohydrate diet is not. While odd-chain fatty acids do increase the need for biotin, they are found in only trace amounts in dairy, fish, and some plants, and the median intake of them is less than a gram per day, far lower than the 20 grams of biotin requiring amino acids that occur in each 100 grams of non-collagen protein. While de novo lipogenesis, the conversion of carbohydrate to fat, does require biotin for the acetyl-CoA carboxylase reaction, it is generally a minor pathway in humans. Outside of the exceptionally rare event where someone's total carbohydrate intake exceeds their total caloric requirement, perhaps a hot dog bun eating contest or a tribal fattening ritual, it ranges 2 to 10 grams per day. It's highest in those who are obese during the follicular phase of a woman's menstrual cycle, or in those who consume approximately 60% of calories as sugar. While obesity and a high sugar diet are likely to meaningfully tax the biotin requirement, and while the biotin requirement is likely to go up meaningfully in the follicular phase of a woman's menstrual cycle, the most these factors will add in total is generally about an extra 8 grams of fat synthesized. This is meaningful, but much less than the effect of eating extra protein. It would be more precise to adjust the gram for gram comparisons for the size of each molecule involved, but it wouldn't change the conclusion that a high protein diet has much more potential to tax biotin status than a high carbohydrate or high fat diet. It follows from this that the amount of protein in the diet is one of the main determinants of the biotin requirement, and the biotin requirement is probably best expressed per 100 grams of protein per day. But how much do we need? Before we can answer that question, we need to understand the best markers of biotin status, because if we don't understand the markers of biotin status, how are we supposed to measure the biotin requirement? So, what is the best marker of biotin status? Studies that attempt to determine the biotin requirement use a variety of markers. In lymphocytes, measurements include the activity of biotin-dependent enzymes, the degree to which their activity increases after adding more biotin, or the concentration of enzymes that are verified to be bound to biotin. In blood or urine, measurements include biotin, breakdown products of biotin, or metabolic byproducts that arise as a consequence of biotin deficiency. Chief among these is the abnormal leucine metabolite 3-hydroxyisovalerik acid, sometimes abbreviated 3-HIA, as well as its carnitine conjugated counterpart 3-hydroxyisovaleryl carnitine. Often the studies induced biotin deficiency experimentally by feeding spray-dried egg white dissolved in a drink such as orange juice. It is important to remember that 3-HIA is conjugated to carnitine precisely because it is a toxic, abnormal byproduct of leucine metabolism that the body tries to get rid of to defend its own energy metabolism. While it is possible that there is always some basal level of this metabolite in the urine that no amount of biotin will make go away, we have to be open to the possibility that the optimal concentration of it is 0. At minimum, we should be interested in the amount of biotin required to maximally suppress it. So here's a rundown of what studies have shown on the utility of these different markers. 10 adults, 8 of whom were women, consumed enough egg white to bind 7 to 8 times the biotin in their diets for 28 days. The only markers that reached 100% sensitivity, meaning they captured everyone with no false negatives, were lymphocyte propionyl-CoA carboxylase activity and urinary 3-HIA at the end of the 4th week. 2 weeks in, urinary 3-hydroxyisobutyryl carnitine was actually the best marker, but it never reached 100% sensitivity. Plasma levels of 3-hydroxyisobutyryl carnitine work similarly to urinary concentrations, but they also do not reach 100% sensitivity. A larger number of similarly small studies found that urinary 3-hydroxyisobutyrate acid is not perfect. With a larger sample size, it captures about 90% of people made biotin deficient over the course of a month. One of them showed that only 35% of the subjects developed low serum biotin. Thus, serum biotin is much less valuable than urinary 3-HIA. A later randomized crossover trial gave 7 men and 10 women a biotin-depleted diet, a biotin-sufficient diet, meaning 30 micrograms per day, or a biotin-supplemented diet with 600 micrograms per day. They used roughly half as much Avidin, and their depletion phase was 3 weeks long rather than 4 weeks, as in the previous studies. Urinary 3-HIA only rose above normal in half the subjects. This may have been because their biotin depletion was not strong enough or long enough, or it may have been that the biotin status didn't fully return to baseline between trials, making the crossover design introduce too much variation. They showed that biotin-activated enzymes in lymphocytes were impacted, but they didn't define normal ranges for them or show how sensitive they were. We will revisit some of these studies soon, but the primary conclusion from this data is that if you deplete biotin strongly enough and long enough, the best marker is elevated urinary 3-HIA or 3-hydroxyisobutyric acid, while its carnitine-bound counterpart in blood or urine might be better at catching a more marginal deficiency, and more research is needed to be done on the relative value of biotin-bound carboxylases in lymphocytes. Now, let's look at that mysterious repetition of 30 and 300. There's a very odd repetition of 2 numbers in these studies, 30 and 300 micrograms. 30 micrograms per day became the AI in 1998 based on a 1997 paper from Donald Mock's group. As covered above, the paper provided no basis for a 30 microgram AI. That Mock 1997 paper did not use a loading dose and used egg white for depletion. At the end of the study, they gave everyone 300 micrograms per day for 10 days without explaining why they chose this dose. They didn't measure anything after that, so they seem to have done it for the health of their subjects. Once 30 micrograms was made the AI, a later study from Mock's group used 300 micrograms per day as a loading dose to make sure no one was already deficient in biotin when they started the depletion. They cited 30 micrograms as the AI and justified 300 as being 10 times the AI. That is, 300 is definitely more than enough. In a yet later study, they dropped the loading dose to 30 micrograms, the AI. Similarly, a different study from another group referred to 30 micrograms as sufficient because it is the AI. These 2 numbers actually have tremendous significance, even though they are never clearly explained in any of these papers. The FDA's daily values used for food packaging had biotin pegged at 300 micrograms per day until 2016 when it was changed to, guess, drum roll, 30 micrograms per day. Their original daily value was issued in 1993 and was based on the 9th edition of the RDAs published in 1980. In 2016, they finally harmonized the food labels with the work the FNB had done since the mid-1990s to set dietary reference intakes, or DRIs, which include the RDAs and the AIs. As a result, they finally changed the daily value from the 300 micrograms that was based on the 1980 RDA to the 30 micrograms based on the 1998 RDA. The 1980 RDAs are not available online, and I don't currently have access to them. However, Mock's 1997 study was the first biotin depletion study done since 1942. In that study by Seidenstricker and colleagues, a diet supplying 133 133 grams of total protein and 139 grams of dehydrated egg white for every 2,000 calories was used to induce biotin deficiency in 4 subjects. Over the course of 11 weeks, they developed off-and-on symptoms of dry or scaly skin. All of them developed hyperesthesia, or increased sensitivity to stimuli, and paresthesia, like pins and needles. Their muscles hurt. They became anemic. They developed stabbing pains on the left side of the chest near their heart. They became depressed and then drowsy. 2 of them developed mild panic disorder. They developed a striking rise in serum cholesterol. And after the 10th week, they became anorexic. The authors experimented, apparently at whim, with injections of 75 to 300 micrograms of biotin per day in 3 of the 4 subjects. 150 micrograms, they wrote, seemed to be the minimal amount required for prompt relief. End quote. It corrected the skin rashes, chest pains, anorexia, and most of the neurological problems in 3 to 5 days. It corrected the cholesterol within a week. And they don't say by how much, but in an unrelated case report of biotin deficiency, serum cholesterol went from 172 to 745 milligrams per deciliter. That is a striking increase in serum cholesterol. Urinary excretion of biotin was 3 to 5 micrograms per day during deficiency, 50 micrograms per day when receiving the 150 microgram injections, and 140 to 150 micrograms when the authors increased the injected dose to 300 micrograms per day. This suggested that the body can accumulate some 100 to 150 micrograms of biotin per day, and that the accumulation of at least 100 micrograms per day is needed to recover rapidly from severe deficiency. Since the biotin was injected and little was known at that time about how well biotin is absorbed from the diet, and since there will always be some obligate loss in the urine, it would have been reasonable to postulate a requirement between 150 and 300 micrograms per day. However, the amount needed to prevent deficiency might be considerably smaller than the amount needed to rapidly recover from it. Several case reports of biotin deficiency were also published between 1942 and 1997, 2 of which used milligram amounts of biotin for treatment. The lowest treatment dose was 200 micrograms per day intravenously. Let's look in a little more detail at the studies done since 1997 and keep in mind 2 things. Number 1, 300 micrograms per day was the RDA up until 1998 and was the FDA's daily value until 2016, not some wildly high number researchers pulled out of thin air. And 30 micrograms per day was practically pulled out of thin air by the FNB in 1998. So let's not stay particularly biased to 30 micrograms, and let's not take 300 micrograms off the table. How much biotin do we need? In 2002, Donald Mock's group published a paper looking at biotin deficiency markers after a leucine challenge during depletion and repletion. Before they conducted the main study, which itself was small, they did a much smaller study to decide which dose of leucine to use for the challenge. 1 person was selected from each of the 3 conditions. Normal biotin status on a mixed general diet, which was presumably somewhere between 30 and 70 micrograms per day, deficient made so by feeding egg white, and supplemented using 300 micrograms, the old RDA. Here are the results for 3 HIA excretion at 35 milligrams of leucine per kilogram body weight. You can see the graph in the written version. The results seem obvious. Normal biotin status is deficient. It's not as deficient as deficient biotin status, but there's a tripling of 3-hydroxyisovaleric acid on the leucine challenge, while this marker goes nowhere at all in the person given the supplement. Granted, with an N of 1 in each group, this is not good enough to make any firm conclusions. Still, the dose response is so clear that it is very difficult not to take this seriously until there's better evidence as an effect of the biotin. When this was repeated at 70 milligrams leucine per kilogram body weight, the results were completely consistent. It rose in the supplemented person, but to barely more than 10 millimoles per mole of creatinine, just higher than the 2 measurements taken before and after the lower dose of leucine. In the person with normal status, it rose a little higher than it had with the lower dose of leucine, and in the deficient person, it went through the roof. Leucine is roughly 8 to 12% of animal proteins and 6 to 12% of plant proteins. Taking 10% as typical of animal protein, the lower dose is equivalent to a 70 kilogram adult consuming 24.5 grams of animal protein. The higher dose is the equivalent of 49 grams of protein. We could round those to the nearest 10 and say that these 2 doses of leucine are the equivalent of 25 or 50 grams of protein per meal. Thus, the two doses are pretty fair descriptions of typical protein intakes per meal on the lower and higher sides of the average. They will be absorbed faster when taken as a supplement and probably oxidized at a higher rate. Nevertheless, even at the low dose, normal looks deficient, so normal biotin status is probably inadequate to handle a 50 gram per meal protein load, even if it's whole foods digesting slowly. Another point about this mini experiment is that the two challenges were done sequentially. That means that the supplemented person was continuing to take 300 micrograms per day, while their fasting 3-hydroxyisovaleric acid declined from 6.8 to 1.8. This is consistent with the possibility that 3-hydroxyisovaleric acid is not maximally suppressed on normal biotin intakes, and that 0 should be on the table as a possible optimal urinary concentration. We unfortunately do not have 3-HIA excretion measured in people randomized to different biotin intakes in groups larger than n equals 1. However, we can glean some low confidence inferences by looking at the different values for 24-hour 3-HIA measured in micromoles generated in the depletion-repletion studies from Mock's group. In Mock's 1997 study, there was no loading dose, and baseline 3-HIA among the 11 participants was 115. This was very similar to their 1993 study, in which the 3-HIA for 10 different unsupplemented individuals was 112. In their 2002 study, a 300 microgram per day loading dose for a week, followed by a 1-week washout, led to a baseline mean of 100. After egg white depletion, 2 to 3 weeks of an 80 microgram per day supplement during the repletion phase brought it down to 84. In their 2005 study, they replaced the 300 microgram loading dose with 30 micrograms. This led to a baseline mean 3-HIA of 117. While they did a depletion-repletion protocol as part of the study, they did not report the 24-hour 3-HIA during the repletion period. In 2011, finally, they published data from the last two studies side by side. Normalized to creatinine rather than expressed as the total over 24 hours, the urinary 3-HIA was actually double in the study with the lower loading dose, but it wasn't statistically significant due to the high variability. It turned out that the trend in the second study may have been influenced by a participant with undiagnosed heterozygous genetic deficiency of beta-methylcrotoncyl-CoA carboxylase, and they didn't publish any data with that individual removed. Comparing across these studies, we can glean a hint that 30 micrograms per day added on top of typical dietary intake is not enough to move the mean 24-hour 3-HIA below the 112 to 117 range, while 1 week of 300 micrograms per day will bring it down to 100, and 2 to 3 weeks of 80 micrograms will bring it down to 84. If we discount the 30 microgram loading dose on the basis that the finding could have been influenced by the individual with the genetic disorder, the following picture seems to emerge. Typical intakes of biotin, around 50 micrograms per day, result in mean 24-hour urinary 3-HIA somewhere around 115. While adding 80 micrograms per day appears to shave off another 10 points per week from this number, and adding 300 micrograms per day appears to shave off 15 points per week. Since none of these comparisons are randomized, and since we have neither large numbers of people nor statistical tests for comparison, the inferences are weak. However, they are made considerably stronger when we look at a result from the randomized crossover trial published in 2013 by a different group. What we're looking at, and the figure is in the written version, are the concentration of biotin-activated beta-methylcrotoncyl-CoA carboxylase in each of the three groups. 17 subjects consumed a low biotin diet for 2 weeks and then were randomized to one of the 3 different biotin intakes, each 3 weeks long, with 2-week washout periods between them, where the subjects ate whatever they wanted. Egg white drinks were used to deplete biotin in the deficient group. In the sufficient group, egg white drinks and biotin supplements were adjusted to yield 30 micrograms per day of absorbable biotin. The same approach was used for the supplemented group, only yielding 600 micrograms per day. The supplemented group has an 83% greater mean concentration of the biotin-activated enzyme that prevents 3-HIA accumulation. This provides proof of principle that 30 micrograms per day does not maximize the ability to metabolize leucine. It also provides a mechanistic basis strengthening the inferences that intakes above this help reduce 24-hour 3-HIA. Since the studies from Donald Mock's group suggest a time-dependent decrease in this marker, and since no one has tested any supplement for longer than 3 weeks, it remains to be seen whether it can be brought down to 0 or what the point of maximal suppression actually is. Our quantitative inferences are still not strong, but we don't have anything better, so let's see what we can squeeze out of them. If average biotin intakes are 30 to 70 micrograms per day, let's use the middle value of 50. Adding this to the supplemental doses of 30, 80, 300 micrograms yields total intakes of 80, 130, and 350. From the comparison of the studies from Mach's group, raising the total intake from 50 to 80 seems inadequate to bring 3-HIA down in 1 week. 130 micrograms seems to lower it over the course of 2 to 3 weeks. And even though 350 micrograms is 2.7 times greater of an intake, it only works 50% faster. This suggests that 130 micrograms may be nearing the maximum that can be well absorbed, utilized, and retained, at least relative to the needs of the participants in that study. Similarly, with the 2013 paper, the fact that the lymphocyte concentration of biotin-bound beta-methylcrotonil-CoA carboxylase more than doubled when going from deficient to 30 micrograms, but less than doubled when going from 30 to 600, suggests that 600 micrograms is considerably more than needed to maximize the activation of that enzyme. The suggestion that 130 micrograms might be nearing the maximally useful dose is very compatible with the results of the 1942 experiment, suggesting that the maximum that can be retained and used in a day is about 150 micrograms. However, how does protein intake fit into all of this? The biotin requirement is a function of protein intake. In Seidenstricker 1942, the protein content of the experimental diet was 133 grams for every 2,000 calories. The food intake was not quantified, though it obviously decreased as the anorexia set in. The Mach group aimed for 2 grams protein per kilogram body weight during their studies, but they did not analyze the self-selected diets leading up to the baseline measurements or during the repletion periods, and they did not report their subjects' body weights. However, mean protein intakes among Americans in grams per kilogram of ideal body weight were 1.4 to 1.5 for males and 1.1 to 1.2 for females at the time of those studies, circa 1999 to 2000 and 2005. In grams per day, mean protein intakes were 103 to 111 in males and 70 to 75 in females. We don't have enough data to separate biotin requirements by sex, so if we just take the average of these, we wind up with 130 to 150 micrograms per day of biotin being approximately optimal for each 90 grams of protein. Given how loose our estimates are, we could round this to an easier to remember 150 micrograms of biotin for every 100 grams of protein. The fact that in Mach 2022, the leucine equivalent of 25 or 50 grams of protein did elevate 3-HIA in someone with normal biotin intake, but not in someone with 350 micrograms per day, suggests that normal biotin status is not adequate to handle the equivalent protein load. Normal intake was presumably 50 micrograms of biotin and 90 grams of protein per day. The leucine load was equivalent to a single meal on a daily protein intake between 75 and 150 grams of protein. This does not necessarily show that consuming more protein would increase the biotin requirement even further, but it does show that it is very likely to be the case. Consider the following. Very little is known about the regulation of the expression of propionyl-CoA and beta-methylgluconyl-CoA carboxylases. Much of what does exist is either on bacteria, effects of biotin, or focused on gene therapy for the genetic disorders in the two enzymes. However, there are a handful of hints that these enzymes are increased by their own substrates. High concentrations of propionate stimulate its own metabolism in human liver cells. In the bacterium Rhodobacter spheroides, feeding propionate increases its production of propionyl-CoA carboxylase 20-fold. During the differentiation of mouse stem cells into adipocytes, there's a regulatory system that ties the uptake and supply of leucine to increased production of beta-methylgluconyl-CoA carboxylase. And we know that eating more protein increases protein oxidation. Consider the comparisons from this human study. The protein intakes went from 0.8 to 1.8 to 3.8 grams of protein per kilogram body weight. This first increment is 2.2-fold higher, the second is 2.1-fold higher. Leucine oxidation went up 53% in the first increment and 16% in the second. However, whole body total protein oxidation went up 79% in the first increment and 2.1-fold in the second. This was done in 5 male endurance trainers at rest. Incidentally, it was done in the lab of Nancy Rodriguez, who taught the protein section of my macronutrient class during graduate school. They simultaneously measured leucine oxidation with isotopically labeled leucine infusions of whole-body protein oxidation using respiratory gas exchange and urinary nitrogen. This is the only study I was able to find that measured both of these metrics at the same time across large differences in protein intake. There may be others, but I didn't find them at the time that I had to prepare for this. Roughly speaking, each doubling of protein intake doubles protein oxidation. However, leucine oxidation seems to start decelerating at intake somewhere beneath 1.8, whereas total body protein oxidation is accelerating at 3.8. The oxidation of leucine, isoleucine, and valine, the branched-chain amino acids or BCAAs, will tend to begin in muscle, while the oxidation of methionine will mainly begin in the liver. The gap between deceleration in leucine oxidation and the acceleration in total protein oxidation is probably explained by the fact that exercise is the main factor increasing BCAA metabolism in muscle, whereas the need to support whole body energy balance and dispose of excess amino acids takes place mainly in the liver. If the biotin-requiring enzymes of protein metabolism don't increase their expression to support this linear increase in protein oxidation that occurs with increasing dietary protein, then high-protein diets would be catastrophic to metabolism, but they aren't. It stands to reason then that until this is better studied, which may take a very long time, that biotin needs go up in proportion to the metabolism of biotin-requiring amino acids. BCAAs make up about 88% of the biotin-requiring amino acids. If we assume BCAAs follow leucine and methionine follows total protein, and if we smooth out the deceleration of leucine oxidation, the acceleration of total protein oxidation to fit a straight line to generate a simple target rather than one that requires an algebraic formula, we arrive at the following. Number 1. Adding 3 grams of protein per kilogram body weight increases BCAA oxidation 67% and methionine oxidation 3.8-fold. Number 2, weighting BCAA as 88% and methionine as 12%, this represents a 96% increase in the need for biotin. Number 3, per gram protein per kilogram body weight per day, the biotin requirement goes up 32%. 4, normalizing this to a 70 kilogram or 154 pound adult, the biotin requirement goes up 23% for every 50 grams of protein. If we start from a basal requirement of 150 micrograms of biotin on a diet with 100 grams of protein, each additional 50 grams warrants an extra 35 micrograms of biotin. That looks like this. 100 grams of protein, 150 micrograms of biotin. 150 grams of protein, 185 micrograms of biotin. 200 grams of protein, 219 micrograms of biotin. 250 grams of protein, 254 micrograms of biotin. 300 grams of protein, 288 micrograms of biotin. 318 grams of protein, 300 micrograms of biotin. Gelatin is very low in biotin requiring amino acids, so should be excluded from the total protein intake in this calculation. There are no randomized studies showing that protein increases the biotin requirement in this fashion, but protein is the single dietary variable that biochemistry would predict to have a quasi-linear, sizable impact on the biotin requirement. These values are also based on piecing together the findings of very small studies. As such, they should be regarded as ballpark figures that are open to revision with further research. Nevertheless, it is much more important to get enough biotin than it is to be certain of the science behind the target. Notably, this would allow the consumption of 318 grams of non-collagen protein without a biotin target that exceeds the RDA that prevailed until 1998 or the FDA's daily value that prevailed until 2016. The biotin requirements increase in pregnancy and lactation. It should not be surprising that pregnancy is the one condition where the demand for lipid synthesis would outcompete a hot dog bun eating contest or a tribal fattening ritual, since in this case the mother is making a whole new human, and thus the biotin requirement follows. Pregnancy increases 3-HIA excretion by 69% on a controlled biotin intake of 57 micrograms per day. Lactation decreases leucine metabolism but increases biotin loss. These two factors probably cancel each other out to some degree and leave lactation with a similar requirement as pregnancy. Pregnant women lost less of the 3-HIA in the carnitine-bound form than non-pregnant controls, suggesting that carnitine was limiting during pregnancy. While showing that 3-HIA goes up 69% is not the same thing as saying 69% more biotin is required to prevent it from going up, the downside risk of not getting enough biotin is birth defects. Until we have stronger data, I think it is wise to shoot for a base during pregnancy and lactation of 100 grams of non-collagen protein and 255 micrograms of biotin, with 60 additional micrograms of biotin for each 50 grams of non-collagen protein beyond that. Now, were the people in these experiments of the biotin requirement truly deficient? And does it matter? Some people may object that the classical deficiency signs were never observed in the experimental depletion-repletion studies done in the 1990s and later. This was deliberate, since it was considered unethical by that time to induce a crippling deficiency. However, half the subjects in mock 2002 noticed themselves or were told by others that they had, quote, an unusual body odor, which I would interpret as a significant but not very specific sign of a metabolic disorder. I take unusual to mean something different than bad body odor. It is a remark about smelling strange, not bad in a normal way. More importantly, I agree wholeheartedly with this quote from the mock 1997 paper. Quote, for a diagnosis of biotin deficiency, one might insist that the pathognomonic signs and symptoms of deficiency be present and that they respond to biotin supplementation. Indeed, these were the criteria we applied in reporting the first case of biotin deficiency associated with biotin-free parenteral nutrition. However, delaying recognition until that point is not acceptable because nonspecific deleterious effects such as teratogenesis, immune deficiency, neurological symptoms, or growth retardation may precede the appearance of the pathognomonic findings. End quote. Pathognomonic means the telltale textbook signs that differentiate biotin deficiency from other diseases. Relying on the telltale signs of the textbook case of deficiency is deeply problematic. The B vitamins are so interlaced with one another in metabolism that they frequently mimic each other's signs for one, and some of the things you would expect from decreased energy metabolism as would occur in any B vitamin deficiency, poor glucose handling, twitching, fatigue, are wildly nonspecific, which means that by the time you get to anything pathognomonic, you are way past the point of having damaging symptoms. But as I have said several times, including at the outset, because you produce neurotoxins from the protein that you eat when you don't have enough biotin, you cannot have any symptoms at all, and you can still be slowly and steadily degrading your neurological health, riding under the radar for decades if you're not paying attention to optimizing your biochemistry, so no one should go without biochemical optimization. Our goal should be to defend and support health, and that requires a much keener eye toward biochemical optimization than it does toward recognizing and curing exceedingly rare textbook deficiency syndromes. Maximally suppressing 3-HIA and optimizing any other biochemical indices of biotin status we can develop should be the primary goal of setting a biotin target. And when you pit pathognomonic diagnosis against biochemical optimization, I think it's safe to say that conventional medicine ruins everything. Okay, your microbiome will not save you. This paper from last year, linked in the written version, makes what is probably the strongest case to date for intestinal biotin synthesis contributing to biotin status in humans. Here are the key points. Number one, biotin is both synthesized and consumed by gut bacteria. Net biotin synthesis potential is associated with microbiomes dominant in Bacteroides and Proteobacteria, while net biotin consumption is associated with microbiomes dominant in Firmicutes. 2. When humans were independently subdivided by obesity status and diabetic status, there were some changes in proportions of biotin synthesizers and consumers, but from figure 1D of that paper, there doesn't seem to me to be any coherent trend. Number 3. Obese people have lower serum biotin and higher 3-HIA than non-obese people, despite not having lower biotin intakes. And number 4, in mice, an obesity-producing diet lowers biotin synthesizers and raises biotin consumers in the gut. Broad-spectrum antibiotics, fecal microbiota transplants from severely obese humans, and obesity-producing diets all decrease serum biotin in these animals. Conversely, bariatric surgery and prebiotics both increase their biotin status. This sounds like a clean story favoring the contribution of biotin synthesized in the gut to human nutritional status, but not so fast. First, these authors rely on their mouse studies for all of their causal inferences, yet the paper does not even mention the word feces or coprophagia, and the authors took no measures to prevent the mice from eating their own feces. Mice use their forelimbs to take feces directly from their anus, and they put it in their mouth. This is not something they do occasionally. It's something they consistently do every day. They eat more of it when pregnant and lactating and less of it when they get old, suggesting that they purposefully eat more of it when their nutritional status requirements are higher. Successful prevention of this with physical restraints causes pregnant mice to miscarry if their diet is deficient in B vitamins, and studies in a related species, the lab rat, show that they will consume as much as 65% of their own feces, even when kept in wire-bottom cages that are designed to cause the feces to fall through the wires. So, it is abundantly clear that mice get nourished by B vitamins synthesized in their gut, but that's because they actually eat those B vitamins. They eat their feces, which contains the microbes that made the B vitamins, and they digest those microbes in the acidic environment of the stomach and the enzyme-rich environment of their small intestine, releasing biotin and other B vitamins to be absorbed by their small intestine's massive absorptive area. In the paper that I had cited before, the microbes thought to be net biotin synthesizers are defined as such because they do have the genes needed to synthesize biotin, but they lack the gene needed to make a biotin transporter that would be involved in taking up biotin from the environment. But if they lack the transporter, how are they supposed to release biotin to be absorbed by the human gut? You would have to count on them dying and being broken up by the immune system or otherwise digested to release their biotin. But even with that happening, you're missing the acidity of the stomach and the vast absorptive surface area of the small intestine that you would be able to harness if you were to eat your feces like a mouse, which I'm not saying you should, but I'm just saying you don't do it. Alas, I am not any more convinced than you are to try. Obesity may be associated with alterations in the microbiome, but because of its association with insulin resistance, it's also associated with increased de novo lipogenesis and gluconeogenesis, both of which raise the biotin requirement. Type 2 diabetes, which is strongly correlated with obesity, is also characterized by low levels of ATP. ATP is used to create sodium gradients needed for absorption of biotin by the sodium-dependent multivitamin transporter, or SMBT, which is also needed in the kidney to keep it from spilling into the urine. Thus, obesity is likely correlated with increased needs for biotin, lower biotin absorption, and a greater proportion of circulating biotin lost in the urine. We certainly cannot rule out that the microbiome makes a contribution to biotin status in humans. At least some microbes appear to release biotin into their environment because natto is very high in free biotin. Additionally, human colon cells contain transporters capable of absorbing biotin. However, it is simply not possible for the biotin that may be released by some microbes to be potentially absorbed by some expression of biotin transporters in the human colon to reach anywhere near the importance of the biotin in the feces of the mouse who eats it for dinner. My suspicion is that the main role of the human microbiome in influencing biotin status is through inflammatory effects that hurt biotin absorption and hurt insulin signaling, contributing to increased biotin needs. More importantly, it is clear that the microbiome cannot eliminate the dietary requirement for biotin. The facts that biotin deficiency can occur on biotin-free total parenteral nutrition, or TPN, which is food provided intravenously, and on biotin-free infant formula, and that some 130 to 150 micrograms of dietary and supplemental biotin appears to be needed in healthy adults to optimize lutein metabolism, all show that there's a genuine dietary requirement for biotin regardless of what the microbiome may or may not provide. So, now we look at the best biotin-rich foods. In a very extensive methodological appendix that is attached to the written version of this article, but which we're not going into in the video or the podcast, I describe why research into the amount of biotin in foods is essentially in a state of replication crisis, with methodological questions that are in desperate need of resolution. Therefore, all numbers in this section are intended as tentative estimates, possibly subject to a major revision if future research is to finally resolve these issues. This section relies on a paper from Donald Mock's group in America, 2 publications from a group in Japan. And as described in the appendix, I've drawn many insights from the Japanese papers, but have normalized their data to the previous estimates for egg yolks, since their exact numbers seem to be very questionable. Additionally, I'm drawing insights from the fact that old studies looked at the ability of a food to cure a biotin deficiency in animals, and this is a biological assay that is of the highest order in terms of the priority of showing the biological truth of the biotin in that food. And that makes liver, egg yolks, and nutritional yeast be at the top of the list. One thing that I think is that I'm most certain of then is that liver, egg yolks, and nutritional yeast are at the top of the list. One thing that I think is rather clear, fresh meat and fish are usually a decent source, but don't contain enough biotin to cover their own protein. And one thing I'm reasonably confident of is that the best vegetarian source is natto. And if natto is properly made, it can rival liver and egg yolks. I am strongly suspicious that grass-fed pasture-raised animal products are 3 to 4 times higher in biotin than grain-fed confinement-raised animal products. The thing I find most concerning about existing claims are that there are reasons to believe the biotin in peanuts is as unavailable as that in raw egg white. And for now, no one should rely on peanuts for biotin. Until this is further studied, I would also be very cautious about using nuts in general. The Japanese research suggests numerous interesting trends. Natto actually tops the egg yolks. It actually tops egg yolks if it's made from ground soybeans, but falls considerably beneath egg yolks if it's made from large whole soybeans. The principal determinant of how much biotin is found in natto is the amount of surface area in the bean exposed to the fermenting bacteria. Bacillus natto, Acidobacter aceti, and yeast, which are used respectively in natto, vinegar, and yeast beer and bread, all produce biotin. But Lactobacillus casei, which is found in cheddar cheese but measured by this group in a drink called Yakult, does not. Natto has far more than the others. Until more fermenting microbes are measured, this tentatively suggests that we can ferment anything with Bacillus natto to produce an excellent food source of biotin, and that most acetic acid dominant ferments will be good sources of biotin while lactic acid ferments will not be. But we need more research. Milk and all dairy products are 3 to 4 times richer in biotin in the summer than in the winter. And until more research is done on this, I take this to mean that the pasture-raised animal products are probably 3 to 4 times richer in biotin than grain-fed confinement-raised animal products. In rats, kidneys and liver are both rich in biotin, and ovary and testes are most likely rich in biotin based on their own expression of the biotin recycling enzyme biotinidase. Until further research is done, this suggests to me that organ meats in general are likely to be rich in biotin, with liver and kidney being the most reliable sources. People with biotinidase deficiencies benefit from consuming free biotin rather than biocytin or protein-bound biotin. The proportions of biotin in the free state are high in egg yolk and natto, moderate in milk, and low in yeast, and are undetectable in spinach and broccoli. Thus, egg yolk and natto are the best foods for people with biotinidase deficiencies. Here are tentative values for biotin in some of the best food sources, all listed as micrograms biotin per 100 grams, or 3.6 ounces, of food, unless otherwise noted. Egg yolk, 383, which is 21 per egg yolk. Natto, 258. The range is 52 to 60,071. That's not 52,000, but 52 is the lower end, and 60,071 is the top end, and 258 is the average. Chicken liver, 187.2, beef liver, 58, salmon roe, 48, anchovies and olive oil, 10.2, summer milk in Japan, 6.7, salmon, 5.4, hamburger, 4.5, pork chop, 3.4, hot dog, 3.7, strawberries, 1.5, fresh avocado, 0.96, broccoli, 0.94, carrots, 0.62, canned mushrooms, 0.216, cauliflower, 0.16, 2% milk from an American grocery store, 0.11, frozen spinach, 0.7, and canned green beans, 0.007. Above, the figures for natto, salmon roe, summer milk, and anchovies come from the Japanese research, while the others come from the American research. At the current time, I have greater confidence in the numbers for the American research for reasons explained in the appendix in the written version. Below are foods that, according to the Japanese research, have very high amounts but are much more likely to be used in small servings as seasonings or ingredients, and so have their amounts listed per 10 grams. Rice bran, 22.5. Royal jelly, 22.4. Ebios brewer's yeast, 16.9. Pepper, probably black pepper, it's not stated, 15.2. Coffee, 14.7. Makabu seaweed, 3.3. Sake vinegar, 2.5. Soy sauce, 2.5. Garlic, 2.4. Sauerkraut, 2.3. All right, that's kind of complicated and a little bit of a mess in the data, so let's make up something simple. The biotin rule of thumb. Fresh meats have decent amounts of biotin, but not enough to cover their protein. For example, salmon, a relatively high biotin animal protein, will typically have 25 grams of protein per 100 grams. In our model, this adds 17.5 micrograms to the biotin requirement, but adds only 5.4 micrograms of biotin. On the other hand, if you top the salmon with a single egg yolk, you would add another 21 micrograms of biotin while only adding 2.7 grams of protein. The egg yolk's protein carries less than a 2 microgram biotin requirement, leaving more than 19 micrograms to cover the more than 25 grams of protein from any other food. So a simple rule of thumb to balance protein and biotin would be to consume 1 egg yolk or its equivalent for every 25 grams of other non-collagen protein that you eat. Here are several equivalents of an egg yolk. 8 grams of natto, 9 grams of chicken liver, 10 grams of rice bran or royal jelly. Keeping in mind rice bran is high in phytate and convert mineral status. 36 grams of beef liver, 44 grams of salmon roe, 12.5 grams of Ebios brewer's yeast. Keeping in mind live yeast can deplete B vitamins according to some older research, so until we learn more, yeast should be cooked. 14 grams of black pepper, or 14.3 grams of dry coffee, roughly 1.5 standard brew cups. This 1 egg yolk equivalent per 25 grams of non-collagen protein should fall upon a background base of 50 micrograms. In all likelihood, this first 50 micrograms will be met automatically if emphasizing grass-fed animal products, using some fermented foods at each meal, and consuming a large volume of fresh produce. We're almost done, but let's first make a note about egg whites and protein supplements. Egg whites retain 71% of their avidin when poached, 33% when fried, 40% when boiled for 2 minutes, and the avidin is mostly destroyed after boiling for 4 minutes and completely destroyed after boiling for 8 minutes. Thus, the only 2 reliable ways to get rid of avidin are to boil 4 to 8 minutes or throw the egg white in the trash. Each egg white contains enough avidin to approximately bind the biotin in the yolk. This means that if you eat whole eggs, these are how many whole eggs you would need to get the equivalent of 1 egg yolk. 3.5 poached eggs, 1.7 eggs boiled for 2 minutes, 1.5 fried eggs, and 1 egg boiled for 4 to 8 minutes. If you use commercial egg white protein, I would demand a certificate of analysis from the company showing the residual avidin activity of their product. I doubt you'll ever get one. Without that, I would either not use these products at all, or I'd always use them in preparations that I thoroughly cook myself. Notably, eating a diet of whole food proteins with naturally occurring fats and balancing each 100 grams of non-collagen protein with at least 10 grams of collagen would likely put you much closer to 150 grams of protein than to 300. However, it becomes quite easy to push your protein up higher when using protein supplements. If you are using supplements to boost your protein, it should not be surprising if you need supplements to boost your biotin. If you cannot find a way to balance protein supplements with whole food biotin, I believe it is wise to make sure you're meeting the biotin target with a biotin supplement. The best low dose one that I can find is the Life Extension 600 microgram biotin supplement. The bottom line, we desperately need more research into biotin requirements, the biotin content of foods, and the bioavailability and biological value of different foods. Both the biotin targets and biotin content of foods listed in this article, this podcast, or this video, depending on where you're watching and listening, are uncertain and subject to revision as new research comes out. Unfortunately, all signs indicate that this research will not come out fast enough due to the erroneous belief that biotin is nutritionally unimportant because of its ubiquity in foods and its abundant synthesis by gut microbes. My best attempt to hobble together conclusions from the data that exists is as follows. 1. Adults should aim for 150 micrograms of biotin per day on a base of 100 grams of non-collagen protein intake and should increase this by 35 micrograms for every additional 50 grams of non-collagen protein after this. Number 2. The simplest way to achieve this is to make a base diet rich in grass-fed animal products, a diversity of fermented foods, and a large volume of fresh produce. Then add 1 egg yolk equivalent for every 25 grams of non-collagen protein in your diet. Number 3, each egg yolk equivalent can be any one of the following: 1 raw or cooked egg yolk with the white thrown in the trash, 1 whole egg boiled for at least 4 minutes, 1.5 fried eggs, 3.5 poached eggs, 8 grams of natto, 9 grams of chicken liver, or 36 grams of beef liver. More examples are given above under the biotin rule of thumb. Pregnant and lactating women should multiply these values by 1.7. Children should adjust downward based on protein intake. All egg white products should be very thoroughly cooked and none should be consumed as is. Bought from a vendor unless that vendor, good luck, provides a certificate of analysis showing no residual avidin activity. If you supplement protein, supplement biotin. If you have a biotinidase deficiency, egg yolk and natto are your likely best superfoods. If you want more about the methodological problems with the research into the biotin content of foods, go to the link in the description and read the appendix. And remember, this video and this podcast are only available for 48 hours for free. After that, they become available to MasterPass members only. So go to the link in the description to get the written article with full references linked out to the associated research papers, as well as the video and podcast versions forever or for as long as you remain a MasterPass member. All right, I hope you found this useful. Let me know what you thought in the comments.